Hello, everyone. My name is Akiko Iwasaki, and I'm a professor of immunobiology at the Yale University School of Medicine. Today, I'm delighted to tell you about our work on the immune response to SARS-CoV-2, which causes the COVID-19 disease. Share my screen. This work was done at the Yale School of Medicine with a large number of collaborators, and I'm delighted to tell you about it today. So before I go into the human data, I'd like to just briefly discuss the virus that causes COVID-19, SARS-CoV-2. SARS-CoV-2 is a member of the coronavirus family, and the corona really depicts the fact that there is a crown-like structure that you can find on the surface of the virus. And this is really because there are spike proteins that protrude out of the membrane of the coronaviruses that gives it its typical sort of characteristics of this crown-like structure. And these spikes are used to bind to host receptor and enter into the host cell. And this virus family has a large genome size of over 30 kilobases of positive sense single-stranded RNA genome. And it is a member of a large number of different coronavirus family. For the humans, there are these regular seasonal human coronaviruses that cause 30% of all common colds. And there are also these zoonotic coronaviruses, the beta coronaviruses that cause more lethal forms of disease, such as the original SARS virus and the MERS virus, MERS-CoV. So these viruses, as with any other viruses, enter the host cell through binding to host receptor. In the case of SARS-CoV-2, it's the human ACE2, which is utilized by the virus to enter the cell, whereby the virus can inject its genomic RNA, which can be used to replicate more of the copies of the genome, as well as to produce viral proteins, which can then be expressed and assembled into new virions that can then infect neighboring cells. So one of the features of the COVID-19 is that it appears to cause very heterogeneous diseases depending on the host and the host status. And so some people get completely asymptomatic disease from SARS-CoV-2 infection and are not even aware that they've had the virus, whereas others get mild disease with fever and cough, and others get moderate disease, which require hospitalization, and others get more severe disease, which requires sometimes admission to the ICU and ventilator use. And then not only is the symptomatic sort of features different, but there are also these acute disease versus long-term disease that occur, which we call long haulers, who suffer from very long-term disease consequences for weeks and months of various different symptoms, including fatigue and some neurological symptoms, as well as shortness of breath and multiple other rather debilitating diseases. So why does the outcome of COVID-19 differ so much between people? And obviously it has to do with the age of the individual, as well as the sex of the individual, as well as comorbidity, obesity, pregnancy status, and several other features that distinguish the outcome of this disease. For instance, in children, most children have mild to asymptomatic disease, but later on, after a few weeks, they might come down with the MIS-C, the form of disease that we still don't understand how this inflammatory diseases occur in some children and not others. So these features still become very enigmatic, and we don't quite understand the mechanism. But what we know is that the host status and other host factors determine disease outcome. And so today I'm going to focus my talk on the comparison of immune response between the moderate and severe disease patients that are hospitalized. But before I go into that, I'd also like to point out that one of the causes of the heterogeneity of the outcome of this virus may be because of different target organs that are infected by this virus. In addition to the respiratory tract that we all hear about, this virus can cause infection in the intestine, kidney, multiple other epithelial cells in different organs. As well as we've identified a clear case of a brain infection. This is neurons in the frontal cortex of a patient that died of COVID-19 that is clearly stained with antibody against the spike protein, indicating that direct infection of a variety of organs, as well as inflammatory consequences of this virus infection in various organs might lead to this heterogeneity of disease. So I'm going to tell you an example of the type of research that we do. So my lab studies immune response to a variety of different viruses, including SARS-CoV-2. And this study that I'm going to tell you about was made possible by establishment of a biorepository that was set up by Professor Albert Koh in the Yale School of Public Health, along with myself and several other who helped establish this biorepository, which allows collection of biospecimens from patients as well as healthcare workers who are enrolled in our study. So this is called the IMPACT study. And in the first cohort, we recruit patients that come through Yale New Haven Hospital. And this is the inpatient cohort from whom we can collect a variety of the specimens, including blood, saliva, nasopharyngeal, oropharyngeal, as well as feces, urine, and other specimens that in a serial manner, so that we can study the changes that occur in these compartments over time. And we also have two other cohorts. The second cohort is the outpatient cohort, in which mild infections, disease patients are recruited. And again, we can collect various biospecimens from these people who are enrolled. And then the third cohort is an important cohort in which we enrolled healthcare workers that are at the front line of treating COVID patients. And from them, again, we can collect a variety of biospecimens. And this also served another purpose, an important purpose, which was to detect as soon as possible any evidence of infection through PCR analysis of their saliva and the nasopharyngeal swabs. So these cohorts allowed us to really compare between moderate, severe disease in the inpatient, mild disease in outpatient, and healthy control or early exposure controls. So what do we do with these samples? Well, each biospecimen are being studied by multiple investigators at Yale. But what we've done is to collect peripheral blood mononuclear cells from patients that are admitted to the hospital and that are enrolled in our study. And then we analyzed the PBMC for a variety of cell types that are present in the blood, as well as the activation status of various cells, including T cells and B cells. And of course, we also collected plasma for analysis of cytokines, antibodies, and other measurements. So this study really involved 3 separate groups of people. The first group is the uninfected healthcare workers. We had 107 healthcare workers in the study. And then we had moderate patients, patients who were admitted to the hospital but were not in the ICU, and 33 severe patients who were in the ICU. Some of them are on mechanical ventilator and others who died of this infection. And we ranked these patients, we assigned them clinical scores based on the amount of supplemental oxygen a patient required, all the way from no oxygen to varying degrees of oxygen, over 6 liters per minute of oxygen. And then we were able to study immune responses in all of these patients with varying clinical score over time. So one of the things that we looked for is the change in composition of cell types that is found in the peripheral blood, where we isolate these mononuclear cells using a strategy that allows for enrichment of these types of cells here indicated. So if you look at the healthy PBMC, it's well known that vast majority of those cells, cells composition is the T cells, which consist of both CD4 and CD8 T cells that make up 60 to 80% of PBMC. And B cells and K cells, these are other types of lymphocytes that you find in the blood. And monocytes, these are myeloid cells that are circulating in the blood for patrolling potential infections, and dendritic cells, which are a small fraction of the PBMC, important in stimulating adaptive immune responses. Adaptive immune responses are carried out by T cells and B cells, which are specific to the pathogen and which can form memory responses. And because of the fact that we usually throw away granulocytes, which are neutrophils and eosinophils, from the PBMC collection, they usually are not counted as part of the PBMC. However, when we examined COVID-19 patients, what we saw was a relatively dramatic drop in the percentage of the T cells that composed PBMC, and also increase in monocytes, as well as these granulocytes, which are usually too high density to enter PBMC collection, but in this case, they are consisted of these low density neutrophils and eosinophils that kind of were elevated in COVID patients. So overall, we saw differences in the composition of the peripheral blood mononuclear cell types in COVID patients. And this is sort of illustrated here more in a graphic form, where when you measure T cells or monocyte number or percentage number per mil in the peripheral blood, you see that the healthcare workers in green have a higher levels of T cells compared to those that are in moderate in navy here, and severe disease, which are in pink. And inversely, the monocytes are elevated in the severe patients. Neutrophils are coming up in the PBMC collection, and so are eosinophils. These are the granulocytes that usually we don't detect in the peripheral blood. And when we measured levels of different cytokines in the plasma, we see elevation in cytokines such as IL-1, IL-6, IL-18, and so on. And so there's IL-6 in particular, you see that the severe patients had the highest levels of IL-6 circulating, whereas the moderate had also higher than healthcare worker levels. And this is really what you would find in healthy controls. And so we see many of these sort of inflammatory and antiviral cytokines come up in the plasma of the patients that have moderate and severe diseases, whereas the composition of leukocytes change with these disease severity as well. So what's really distinguishing these moderate versus severe cases? One of the clues of this came from this analysis where when we measure viral titers from the nasopharyngeal swabs of patients over time, what we noticed is that patients who had moderate disease were able to ultimately clear the virus. This is on the x-axis is the days from symptom onset. So within about 25 days of hospital stay, moderate patients were able to clear the virus below the level of threshold of detection. Whereas the severe patients had basically elevated levels and maintained the elevated levels of virus that's detectable in the nasopharynx, indicating that this failure to clear the virus may be at the root of severe disease from COVID-19. And when we looked at the so-called antiviral cytokines, the type 1 interferons and type 2 interferons, as well as type 3 interferons. So these are basically cytokines that are secreted in response to virus infection that are usually induce antiviral factors that control the viral replication. But in the case of COVID, what we see is that these cytokines, interferon alpha, for instance, is being driven by the viral load and not the other way around. So the viral load on the x-axis, you can see that there is a positive association between the increasing viral load and the increasing levels of type 1 interferon and type 2 interferon, and TNF and TRAIL. These are other pro-inflammatory factors that we find in the plasma of these patients. So it appears that instead of these interferons controlling the viral load, it's the viral load that is driving the secretion of these plasma levels of antiviral cytokines. And not only that, when we measured the cytokine levels during the first 12 days since symptom onset in patients who died of this infection, what we find is that they have elevated levels of the antiviral cytokine interferon alpha here, as well as IL-1 receptor antagonist, which is indicative of signaling through IL-1 receptor, which is really one of the key inflammatory cytokines that drive cytokine storms in this disease. So instead of these things having positive impact, the elevated levels of these cytokines are associated with mortality from this disease compared to these patients who were discharged home and who recovered ultimately from this infection. So then we wanted to look more broadly, not just the antiviral cytokines and viral load, but just more broadly, the types of immune responses that are engaged in the COVID patients. And this is just unfortunately, I do have to give some introductory slides on immunology, just to kind of give you a sense for how the immune system works to control different types of pathogens. So as I mentioned, the dendritic cells are a key cell type that educate the lymphocytes, such as T cells, to become different types of effector cells. So these naive T cells can either turn into T follicular helper cells that help the B cells secrete antibody. They can also become Th2 cells, which are good for fighting parasite infections. Th1 cells, which are optimal for antiviral and antibacterial response. Th17 cells are useful for antifungal responses. And then these are the inducible regulatory T cells, which are important to dampen a hyperstimulation of immune response against pathogens. So these things are usually not induced all at the same time. For instance, antiviral response really depend on the Th1 cells that are driven by the cytokines that are released by the dendritic cells to educate the T cells into Th1 cells that can secrete interferon gamma. And this is usually what a viral response looks like. So I want you to remember that these are dedicated to fighting different types of infections, and only one of these types are engaged during an infection. However, when we looked at the blood types as well as the cytokines released by the COVID patients, what we saw was that all of these types, type 1, which is the antiviral, type 2, which is the parasite response, and type 3, which is the fungal response, were all being engaged and elevated in patients with severe disease, which is this pink lines here, whereas the moderate patients either had flatline or a slight increase, but not as much as what we see for the severe patients over time. So this is really, it looks like a misfiring of cytokines and immune responses that are normally dedicated towards helping the host fight against parasite infection and fungal infection, being engaged in some way in the COVID patients. So we wanted to take all the different soluble cytokines that we measured in the patients and asked if there's any pattern that can be gleaned from looking at the different types of cytokines that can, distinguish different types of patients. So we carried out an unsupervised clustering analysis based on the cytokines. And what we saw was that there are three clusters of patients that emerged. The first cluster has the most enrichment in these the A type of signature. And so the darker the color, the higher the levels, Z score for the particular cytokines. What you see is that the cluster 1 has an enrichment in the factors that look like they belong to tissue repair growth factors, such as growth factors for such as VEGF, PDGF, EGF. These are growth factors that allow repair of various different cell types in a tissue, including IL-7, which is a growth factor for lymphocytes. So this signature, the first signature, appeared to kind of indicate reparatory function of the immune system. The second cluster of patients had, in addition, elevated signature for this and this, which we are calling mostly chemokines, which are factors that allow cell recruitment to various tissues. And this is the fourth signature. We're calling it mixed signature because it contains cytokines that belong to all different three different types of immune responses that I mentioned earlier. And then there's the third cluster of patients, which are a handful of patients that really have a highly enriched a profile for cytokines that belong to type 2 and 3 and mixed cytokine signatures. So what do these clusters of patients mean? Well, it turns out that the patients in each cluster have very distinct disease trajectories. So in the cluster 1 patient that had the tissue repair growth factors, they eventually become better from this disease. So the clinical score goes up a little bit, but then they eventually recover and are discharged from the hospital to go home. Whereas cluster 2 and 3 patients have a increase in disease score over time and associated with higher levels of coagulopathy as well as mortality compared to the first cluster of patients. So just based on these cytokines alone, we are able to define different sets of patients that go on to recover versus that go on to severe disease. And so taking all the factors that we measured, we were able to come to some biomarkers for mortality. And one of the highest scored biomarker for mortality turns out to be IL-18, which is really the product of inflammasomes, which are this highly toxic type of inflammatory responses that are engaged in severe patients. And as I mentioned, this antiviral cytokine, interferon alpha, is the second highest biomarker for mortality. And then you can go down the list, and some of these things are more protective, such as T cell activation. So we were able to get a sense for the different kinds of cytokines and cell types that predict either mortality or recovery from this disease. And so to conclude these results, what we found was that the viral persistence in the severe patients distinguish those from the moderate patients that ultimately clear the virus. And it's the viral load that's driving cytokine responses. And increases in these antiviral cytokines actually are associated with mortality within the first 12 days of symptom. And the 4 different signatures of cytokines that we find in the plasma of patients are able to cluster patients into those that recover from infection versus those that go on to develop worse disease. And so if you think about different models of infection, these are the 2 extreme sort of ends of the spectrum of viral infection and antiviral factor secretion. And in this scenario, the viral load, this x-axis is over time. So the patients start to replicate the virus, but then quickly the host responds by secreting interferons that will interfere with the virus replication, and the host recovers with mild disease. Whereas in the case of our hospitalized patients, what we're seeing is that the interferon is coming up, but much later and at lower levels. And the viral load, as a consequence, can replicate a lot faster and for a prolonged period in these patients. And so really the interferon response is unfortunately too little, too late. And not only that, it may be driving the pathological changes that we see in the severe disease cases. So just by looking at these longitudinal immune responses in patients, we're able to kind of model the time course for viral load and interferon response, and really reveal that this second model may be reflecting severe COVID disease. And we can do this kind of models for multiple different factors, not just antiviral cytokines, but for T cells, antibodies, and a variety of other measures. And so this is when we had a large number of data set that we have collected from these patients. And the first analysis that I just told you is really the tip of the iceberg of what we can do with our data set. And so at this point, you know, for immunologists like myself, we were kind of burdened with lots of data from different patients at very different time points. And we wanted to understand whether we can assign importance of various immune cellular state to mortality. So I told you that measurement of cytokines can sometimes predict this mortality. But when it comes to immune cells, there are so many different types, so many different states. How do we deal with such large data? And so I contacted my wonderful colleague, Dr. Smita Krishnaswamy. And she's, as you know, is a computational biologist who also does a lot of immunological analyses. And so she said, yes, we can use this new tool that her lab created, which is called multi-scale fate, where she can combine 2 powerful algorithms, diffusion condensation for multigranular analysis and fate for dimensional reduction, to produce like visualization of different cluster on all levels of granularity. This is really amazing. You would have to refer to her for how she does this, but as a biologist, we really benefited from this multi-scale fate analysis. And so this question about assigning importance to various immune cell states for mortality, a wonderful graduate student in her laboratory, Manik Kuchuru, was able to take all the different flow cytometry data and use the multi-scale fate and found that different cell types really have a different a level of association with mortality. And so what he found, so I asked him to plot all the different cell types, and there's many more. I just shrunk them into something that can fit in the screen, of different PBMC clusters, B cell subsets, different CD8 T cells, and different CD4 T cell subsets. And yes, within each of these lymphocyte subset, there are differences in mortality associated with, let's say, pathogenic Th17 versus Th2, and also different cytotoxic T cells are associated with different mortality scores. So the y-axis really, the higher the y-axis score, the more association with mortality there is per particular cell type. And of course, B cell subsets also have similar types of variation. But what was striking to me is that this analysis really came out with the granulocytes being the number one associated mortality factor of all the different leukocytes, as well, followed by the inflammatory monocytes. And so it's not the lymphocytes that are really highly associated with mortality, but it's rather the granulocytes that we detect, which I told you is highly elevated in the severe patients, that is also really driving the mortality. And these granulocytes, subsetting of these granulocytes by Manic reveal that it's the sort of activated neutrophils and eosinophils that account for this high levels of mortality. So this is a wonderful example of how a biologist laboratory can collaborate with computer science laboratory to really kind of reveal the underlying biology of different cell types that are associated with mortality. And I'm sure Smita's lab can do this for many other measurements other than just mortality. But it's just to give you an example of what can be done when we collaborate together on these analysis of biological data set. So I will this is my final thoughts, how computer science can help. There are many, many ways that biologists and computer scientists can work together to reveal different aspects of what we're doing. But I think beyond genetics, hopefully our collaboration illustrated the fact that biology is really full of phenotypes that are not governed necessarily by the genetic factors. It's everything from metabolism to diet, microbiome, virome, many different aspects other than the genetics can drive phenotype. So by looking at the sort of cellular activation status, for example, and applying some of the computational tools, we can really go way beyond genetics to look at phenotypes and causality. Each patient, as I mentioned, has now accumulated a large number of data, clinical readout and biological readout. So there's like 130 different parameters associated per time point per patient. And we have over a few hundred patients that are accumulated by now. And so this is a real wealth of data that can be mined to understand the sequence of events, because this is a longitudinal analysis. And we would love to learn what causes what is driving what type of outcome. Is the viral load really driving these interferon, and how does interferon cause disease? I mean, I think all of those types of information is hidden in the data set that we have. We just need your help to analyze these things. And we also need a better tool to fill in the gaps in data. So not every patient can be recruited immediately upon arrival. And even if they are recruited immediately upon arrival, we're missing the first week of infection because they're not coming to the hospital. This virus has a long incubation period, and most people aren't coming to the hospital until about a week after exposure to this virus. So how do we fill in the unknown? And we can go back in time. So is there any way that we can sort of impute some of these missing data using computational tool. And we also need unbiased approach to determine biological patterns behind different disease outcomes. As I mentioned, there is the acute disease, the long hauler, the children with this multi-inflammatory syndrome, and COVID toes, and so on and so forth. This virus causes so many different heterogeneous disease outcomes. And how do we mine our data to reveal what is causing these different types of outcomes? And yes, so ultimately, we really need to untangle biological data to reveal best clinical intervention targets. So we see all these things elevated, and they're all correlated with mortality. What is the best target for clinical intervention? Again, it would be great to have computational tools to analyze those things. So I'm going to end here by thanking my amazing lab members who worked around the clock, really, still working very, very hard to not only understand immune response to COVID-19, but also to help implementation of testing. And we actually did a lot of PCR for the healthcare workers to detect early infection. And this impact team that I mentioned before, without whom none of this work can be done. And then Smita's lab, and Smita herself and her lab members who did this amazing work on the multi-scale fate. And there are many, many others that are ongoing that we would love to report soon. And the work that I described was published by multiple authors that are indicated here. And I thank the funding agencies. Thank you so much for your attention.